Salmonella is one of the most common bacterial foodborne illnesses. There are 1.41 million cases and over 500 human deaths per year due to salmonella. Salmonella is a rod-shaped bacteria that utilizes quorum sensing like many other bacteria. Quorum sensing is used to produce many responses in coordination to the population of other bacteria around it. Salmonella invade cells and take them over by rapid replication, which is how they infect the body.
For the cell signaling pathway, the salmonella first approaches the cell. The cell is usually an intestinal epithelial cell. Once the salmonella gets close to the cell membrane, it will release effective proteins known as SIPA and SIPC that do a sort of quorum sensing and detect the receptors TLR2 and TLR4. Once the receptors see the salmonella, they will immediately bind to it and engulf the salmonella into the cell, similar to the process of phagocytosis. Once it is engulfed into a cell, a bubble-like structure forms which protects the salmonella cell from being attacked by the immune system and macrophages. The salmonella will once again release these effective proteins to do another sense of quorum sensing to sense environmental factors such as pH. Usually when it enters the cell, it is a neutral pH. However, by releasing the TLR receptors, the salmonella is able to change the pH to make it an acidic concentration. Once it is acidic, the salmonella is able to copy, grow, and reproduce in this bubble because it is protected from the immune system. Once the salmonella in the bubble has exceeded its maximum capacity, the bubble will burst, and the cells will eventually leave the cell and initiate responses in the body. The cells, because there's such a great quantity, even if macrophages are attacking it, there's too much of an abundance, so the salmonella is able to leave and go through like the intestinal lumen, which is why diarrhea is one of the responses toward this pathway. It also can initiate the inflammatory response, which initiates pain and will open the intestinal cells. By doing this, salmonella can also leave and spread to other parts of the body, and the pain is usually the first time a human might notice they even had the infection. One other response includes apoptosis.
One way that salmonella can be stopped by the body is if the cell develops an irregular shape. If it is not the correct shape, the bubble-like layer can't form around it because it won't be able to sense environmental changes such as the pH level. If the bubble does not form, the macrophage cells will be able to engulf the premature salmonella cells. Since they're killed before they can be dispersed, no infection of the body will occur.
The current direction of research has scientists been looking at bacteria, Salmonella being the number one tested bacteria, because these have a tendency, they have the phenotype that can target tumors and possibly destroy them. What happens is by binding to a receptor, they are able to go inside these cancer cells and still copy and replicate inside these tumor cells because, like their bubble, they are protected from the immune system. So this idea is known as the bacterial therapies and is being used as another way to treat cancer cells. Also, according to Barbara Wang, because the bacteria is living inside these cancer cells, it is slowly destroying them, and a patient's life is said to be doubled.